May I request everyone to kindly keep your mobile phones on silent mode to ensure the smooth conduct of the program.
Good morning to all respected dignitaries, invited guests, faculty members and students. A warm welcome to each one of you to the E.K. Janaki Ammal Memorial Lecture organized as a prelude to the 38th Kerala Science Congress. The Kerala Science Congress, organized annually by the Kerala State Council for Science, Technology and Environment, continues to be a premier platform for scientific exchanges and interdisciplinary dialogue. This year, the 38th Kerala Science Congress will be held from 30th January to 2nd February 2026 at St. Albert's College, Ernakulam.
As part of this year's academic programs, 9 memorial lectures have been instituted to honor eminent luminaries in science, 7 of which are being conducted as preludes to the main event at premier institutions in the state. Today, we gather for the E.K. Janaki Ammal Memorial Lecture, paying tribute to E.K. Janaki Ammal, a renowned Indian botanist who made significant contributions to plant cytogenetics.
Before we proceed to the formal ceremony, there's a short video showcasing the initiatives of KSCSTE.
Kerala State Council for Science, Technology and Environment, popularly known as KSCSTE, was constituted in November 2002 to encourage and promote science technology related activities in Kerala State. It was done by reconstructing the erstwhile State Committee on Science, Technology and Environment, which was established in 1972 in concurrence with the science policy of the Government of India. Over the past 2 decades, KSCSTE spearhead the science and technology activities of the state in general and its own R&D centers in particular. With its headquarters in Thiruvananthapuram, 7 R&D centers, 3 grant-in-aid institutions, 2 centers of excellence and 3 science parks are functional under the council. Our R&D centers are Center for Water Resource Development and Management, Kundamangalam, Kozhikode, Malabar Botanical Garden and Institute of Plant Sciences, Pukkunnu, Kozhikode, Kerala Forest Research Institute, Pichi, Thrissur, Institute of Climate Change Studies, Kannikuri, Kottayam, Srinivasa Ramanujan Institute of Basic Sciences, Vellur, Kottayam, Jawaharlal Nehru Tropical Botanic Garden and Research Institute, Palur, Thiruvananthapuram, National Transportation Planning and Research Center, Ernakulam, Thiruvananthapuram, Sophisticated Test and Instrumentation Center at CUSAT, Kochi, Community Agro-Biodiversity Center at M.S. Swaminathan Research Foundation, Wayanad, Integrated Rural Technology Center at Palakkad are the grant-in-aid institutions of KSCSTE. KSCSTE has also facilitated the establishment of Sri Chitra Tirunal Institute of Medical Science and Technology, Rajiv Gandhi Center for Biotechnology, National Center for Earth Science Studies, and Institute of Advanced Virology. At the headquarters, we initiate several programs to advance the science and technology objectives of the state. Programs for students include Scheme for Promoting Young Talents in Science, Student Project Scheme, and the Pratibha Scholarship for UG and Integrated PG students. For researchers, we have KSCSTE Research Fellowship for PhD scholars, Postdoctoral Fellowship for Post-PhD research, Partnering Academic and Industrial Research Scheme to promote translational research through academic-industry linkage. Our programs for mid-career researchers include Ecology and Environment Program, Science Research Scheme, and Engineering and Technology Program. Programs for women in science include Back to Lab Research and Postdoctoral Fellowship. These programs support women researchers with a break in career. KSCSTE also operates an entrepreneurship development program for SC/ST women in STEM. To support R&D initiatives from superannuated scientists, we operate the Emeritus Scientist Scheme. Selective augmentation of research and development and Shastra Bhoshani, science labs for government high schools in Kerala are 2 of our science infrastructure development programs. Under Shrestha program, KSCSTE nurtures Center of Excellence for World-Class Research in various domains. Our programs for technology development include APJ Abdul Kalam Youth Challenge Program, Rural Technology Program, Technology Development and Adaptation Program, and Engineering Support and Coordinated Research for Specially Talented Children. We initiate environmental conservation in the state by Environmental Information Awareness Capacity Building and Livelihood Program, National Green Corps, and Wetland Technical Unit. The Kerala Biotechnology Commission supports biotechnology development programs, which include biotechnology research support, schemes for industrial collaboration, and entrepreneurship development in biotechnology, Young Investigators Program in Biotechnology, Kerala Biotechnology Re-Entry Fellowship, Biotechnology Innovations for Rural Development, and our capacity building initiatives, notably the DBT Skill Vigyan Program and Human Resource Development in Biotechnology. Activities of the Patent Information Cell and the Intellectual Property Rights Information Centre Kerala facilitate IP awareness and patent-related activities. KSC-STE Scheduled Caste and Scheduled Tribe Cell is a DST-supported project that provides S&T solutions for the development of SC-ST communities. KSC-STE Shastra Puraskaram, Kerala State Young Scientist Award and Science Literature Awards are notable science and technology awards instituted by the Council. KSC-STE is honored to recognize legendary scientists and science advocates through the Kerala Shastra Puraskaram. The National Children's Science Congress, Technology Festival, and Rural Innovators Meet are other important science popularization programs. KSC-STE also supports the observation of National Mathematics Day and National Science Day programs in the state. Kerala Science Park is a new initiative facilitated by KSC-STE for R&D development in the state. Centers of Excellence in Microbiome and Nutraceuticals are the two new initiatives facilitated by KSC-STE. Kerala Science Congress is an annual event of KSC-STE. The event includes 9 memorial lectures to honor the S&T visionaries and to cherish their contributions. Focal theme sessions, technical sessions bring together researchers in various domains. National Science Exhibition is another highlight of the Kerala Science Congress. Sessions like Walk with Scientists, SciSoul, PG Interaction Session are also organized as part of the event. The 36th edition of Kerala Science Congress evinces the presence of 2022 Nobel laureate in chemistry, Professor Morton P. Meldal, addressing the gathering. A celebration of science, innovation, and collaboration, the Kerala Science Congress has, through its many editions, united Nobel laureates, eminent scientists, and emerging researchers. It stands as a testament to Kerala's commitment to advancing knowledge, inspiring generations, and driving discovery for a better future. The 38th edition of Kerala Science Congress is scheduled from January 30 to February 2, 2026 at St. Albert's College, Ernakulam, with the focal theme, Ocean Science for Blue Economy and Ecosystem Restoration. Kerala State Council for Science, Technology and Environment, advocating science and technology innovation in service of the state of Kerala.
It is now my privilege to invite our distinguished dignitaries to the dais. Dr. Anu Joseph, Principal, St. Teresa's College, Ernakulam. Professor A. Sabu, Member Secretary, KSCSTE and General Convener, 30th Kerala Science Congress. Sri Arjun Prasad, Scientist, KSCSTE. Ms. I.K. Nishida, Assistant Professor, Department of Botany and Center for Research, St. Teresa's College, Ernakulam. Professor Usha Vijayraghavan, Professor, Department of Microbiology and Cell Biology, Indian Institute of Science, Bengaluru. Dr. Himashri P.R., Junior Scientist, KSCSTE, National Transportation Planning and Research Center. May I request the dignitaries to kindly occupy their seats on the dais.
To commence the formal proceedings, I invite Dr. Anu Joseph, Principal, St. Teresa's College, Ernakulam, to deliver the welcome address.
A very warm good morning to all gathered here, respected dignitaries on the dais, Honorable Member Secretary of KSCST, Professor A. Sabu, General Convener of Kerala Science Congress and President of the function, Professor Dr. Usha Vijayraghavan, our distinguished speaker of the day, Shri Arjun Prasad, Scientist, KSCSTE, Dr. Himashri P.R., Junior Scientist, KSCST, NATPAC, Srimati I.K. Nishita, Head of the Department of Botany, St. Teresa's College, and esteemed faculty members, students, and dear participants. It gives me immense pleasure to extend a warm and heartfelt welcome to all present here to Janaki Ammal Memorial Lecture, organized by the Kerala State Council for Science, Technology and Environment in association with St. Teresa's College Autonomous Ernakulam as a prelude to the 30th Kerala Science Congress. We are deeply honored that our institution has been chosen as a venue for this prestigious memorial lecture that commemorates the life and legacy of Dr. E.K. Janaki Ammal, one of India's most illustrious scientists and a pioneering woman in plant cytogenetics. Her scientific contributions, her commitment to biodiversity conservation, and her courage to challenge social and academic barriers continue to inspire generations of scientists, especially women in science. Remembering her today is a reaffirmation of our collective commitment to scientific excellence and social responsibility. I extend a warm welcome to Professor A. Sabu, Member Secretary, KSCSTE, whose leadership and vision have been instrumental in strengthening the scientific ecosystem of Kerala. We are grateful to KSCST for its constant support to academic institutions and for creating platforms that bring together science, society, and young minds. A warm welcome to you, sir. It is a matter of great pride for us to welcome today's distinguished speaker, Professor Usha Vijayraghavan, Professor at the Department of Microbiology and Cell Biology, Indian Institute of Science, Bengaluru, an internationally acclaimed molecular biologist and an inspiring and aspiring academic leader. Professor Vijayraghavan's contributions to plant developmental biology and gene regulation have scientifically advanced the understanding of plant sciences. We are indeed privileged to learn from her scholarship and experience. A hearty welcome to you, ma'am. I also extend A warm welcome to Dr. Arjun Prasad, scientist at KSCST, who coordinated the event from KSCST end. Dr. Arjun's work lies at the intersection of science education, technology, and public policy. His research focuses on implementation science, particularly on understanding how scientific ideas, programs, and policies are translated into practice by implementers and policymakers. We are pleased to have him with us for this academic deliberation, and I extend a warm welcome, sir. Dr. Himashri PR, junior scientist at KSST NATPAC, is also with us here. She's a scientist in the Highway Engineering Division and is an active researcher with strong academic credentials. I take this opportunity to extend a cordial and hearty welcome to you, ma'am. I would also like to welcome the head of the Department of Botany and convener of this program, Srimati I.K. Nishita, whose dedicated efforts have been instrumental in organizing this event. A warm welcome to you, Nishita. I would also like to welcome all the other officials from KSCST, NATPAC, and academicians and other delegates who are all present with us here today. I also extend a warm welcome to Dr. Helvin Vincent, head of the Department of Zoology and co-convener of this program, for her valuable support and role in planning and coordinating this event. I also heartily welcome all the research scholars, faculty, students who have gathered here today. Your presence reflects the shared enthusiasm for science and dialogue that events like this nurture. Hosting this memorial lecture aligns perfectly with our institutional vision of empowering young minds, promoting scientific temper, and celebrating role models who have shaped the course of Indian science. Once again, I extend a hearty welcome to all and wish this memorial lecture every success. I'm confident that the deliberations today will be intellectually enriching and truly inspiring. Thank you. Have a meaningful and enriching session ahead.
Thank you, ma'am. May I now invite Professor A. Sabu, Member Secretary, KSCST, to deliver the presidential address.
I don't need. Perfect now. A very good morning, one and all. Most respected Professor Usha Vijayraghavan, Dean, Indian Institute of Science, Bengaluru, my colleagues from the St. Teresa's College and Kerala State Council for Science, Technology and Environment, Dr. Anu Joseph, Ms. I.K. Nishida, Arjun, and Himashri, eminent personalities of the dais, researchers, and my dear students. This program, E.K. Janaki Ammal Memorial Oration, is a prelude to the Kerala Science Congress that we are organizing every year. And the city of Cochin is getting ready to host the 38th session of Kerala Science Congress, starting from 30th of January to 2nd of February. St. Albert's College, Ernakulam again, another important academic institution in Cochin, is hosting 38th session of Kerala Science Congress. As rightly pointed out here, E.K. Janaki Ammal Memorial Oration is one of the important memorial lecture being organized as a prelude to Kerala Science Congress. In fact, there are 9 memorial orations. 2 orations are taking place at the conference venue itself, and the rest 7 is being organized in different academic institutions, universities throughout the state. Day after tomorrow, the Professor P.R. Pisharedi Memorial Oration will take place at Cochin University of Science and Technology. Dr. Rajarajan, Director of the Vikram Sarabhai Space Center, will be giving the memorial oration, P.R. Pisharedi Memorial Oration. The day is very important for KSCST because we have a great scientist with us, a great women scientist with us, Dr. Usha Vijayraghavan. She will be rightly introduced by my colleague, Dr. Arjun Prasad. In fact, KSCST is very much connected to Bangalore Indian Institute of Science. Some of the very senior professors from IISC were always helping KSCST to shape the scientific scenario of Kerala, whether it is Professor K.P. Gobinathan or Professor M. Vijayan, and currently Professor Belram and Dr. Sadish C. Raghavan are some of them who are always with us, with the KSCST, to support our endeavors in spreading science and technology in the state. If you look at the national scenario of state science and technology councils, Kerala State Council for Science, Technology and Environment is rated as the topmost council. We are operating with a budget of 200 crores with the 10 institutions and centers of excellence operating throughout the states. And the science park projects are special programs which comes around a budget of more than 1,000 crores supported by Kerala Infrastructure Investment Fund Board, the KIFBI. So in the coming years, what KSCST is planning is to translate the know-how from the laboratories to processes and products. That's why we are going to have the 3 science park right from Kannur to Cochin and to Trivandrum. Translation of the know-how generated from the laboratories are very much important to develop Kerala as a knowledge-based economy. I'm not taking your time. I told you the Science Congress, the 38th session is happening in Cochin. St. Albert's College is very near to St. Teresa's College. St. Teresa's College is just completed their 100 years. They celebrated their century. The Prime Minister, President, all dignitaries, all democratic leaders in the country visited this prestigious college, having rated with the NAC A++. A college which is giving place for more than 4,000 students to learn, and more than 50% of the students are getting placement. This is more important. And Alberts is again another autonomous college in Cochin, and they are celebrating their 80th year of existence. Hopefully in the coming years we'll be able to organize one of the sessions of Kerala Science Congress in St. Teresa's College also. The registration process for the Kerala Science Congress is coming to an end by tomorrow. So far, 850 research papers were submitted. The evaluation process is going on. There were 1,200 registered delegates attending this session, along with the postgraduate student representatives, school kids, dignitaries, senior researchers, and all. There's going to be a gathering of more than 2,000 delegates during the 38th session of Kerala Science Congress. The National Science Expo that is being organized as part of the Kerala Science Congress is open to all aspirants of science and technology. So I request all the students and researchers from St. Teresa's College to make use of this opportunity and spend some time at the National Science Expo, where 180 stalls are being set up from institutions starting from ISRO and industry R&D segments, and even from startups and young innovators. So, Madam, this is a great opportunity for KSCST to host you here in St. Teresa's College to give the Janaki Ammal Memorial Lecture. Thank you, Professor Anu Joseph and Professor Sajimol, my good friend and former principal of St. Teresa's College, for making arrangements for organizing this program here. Thanks to the leadership of Dr. Nishida and Dr. Helbin for organizing this program in a wonderful way. I hope we will have more collaborations in the future for spreading the message of science to each and every corner of the state. I would like to mention one more thing. This year's Kerala Science Congress, the 38th edition of Kerala Science Congress, is chaired by another eminent professor from the Bengaluru Institute of Science, Professor P. Belram. He is also, though he is not from Kerala, he is also very much associated with the Kerala State Council for Science, Technology, and Environment. So, wishing you all a fruitful session this morning. I hope the younger generation will make use of this opportunity interacting with Professor Usha Vijayraghavan, one of the eminent molecular biologists of our country. So, wishing you the very best and thanking all the faculty colleagues and principal from this college for supporting KSCST for organizing this function. Thank you very much.
Thank you, sir. We shall now have a brief introduction to the life and contributions of Dr. E.K. Janaki Ammal. I kindly request Sri Arjun Prasad, scientist, KSCSTE, to present the same.
Good morning to one and all. President of today's function, Professor A. Sabu, Member Secretary, Kerala State Council for Science, Technology and Environment. Distinguished speaker for today's memorial lecture, Professor Usha Vijayraghavan. Principal of this college, Dr. Anu Joseph. Ms. Nishita, Head and Assistant Professor, Department of Botany, St. Teresa's College. Dr. Himasri, Junior Scientist, NATPAC. Distinguished faculty members and dear students. It is both a privilege and a deeply meaningful moment for us to gather today at this prestigious institution for the E.K. Janaki Ammal Memorial Lecture. This lecture is not merely a tribute to an outstanding scientist. It's a celebration of courage, intellectual freedom, perseverance, and the quiet strength of a woman who chose science as her life's mission at a time when such choices were very rare, difficult, and often discouraged. Dr. Edavalat Kakkad Janaki Ammal was born in 1897 in Telicheri, the present Talasheri, in the Kannur district of Kerala. She stands as one of the most remarkable figures in the history of Indian science. She was a pioneering botanist, a cytogeneticist of international repute, and above all, a woman who lived life on her terms, guided by curiosity, integrity, and a deep love for plants and nature. For the young women gathered here today, her life holds a powerful message. Your background does not define your boundaries, and your gender does not limit your dreams. Dr. Janaki Ammal was born into a time when women's education itself was considered a luxury. Literacy among Indian women was extremely low, and higher education, especially in science, was also unimaginable for most. Yet, supported by an enlightened family and driven by her own determination, she pursued education relentlessly. From Queen's Mary College and Presidency College in Madras to the University of Michigan in the United States, she carved a path that few Indian women had walked before her. She became the first Indian woman to earn a doctorate in botany in the US, and later one of the very few Asian women of her time to receive a doctorate in science. But her journey was never about collecting degrees or titles. It was about doing meaningful science. Dr. Janaki Ammal's scientific contributions were both deep and wide-ranging. At the Sugarcane Breeding Institute in Coimbatore, she laid the foundations of cytogenetic research in sugarcane. Through painstaking experiments and interspecific and intergeneric hybridization, she helped improve indigenous sugarcane varieties, work which contributed directly to India's agricultural self-reliance. It is for this reason that she is often remembered as the woman who sweetened India's sugarcane. Later, at the Johns Innes Horticultural Institution in England, she co-authored the landmark Chromosome Atlas of Cultivated Plants with Cyril Dean Darlington. This work transformed the way scientists understood the genetics, evolution, and breeding of economically important plants across the world. Even during the dark years of Second World War, while London faced mighty bombings, Dr. Janaki Ammal continued her research with extraordinary resolve, an image that powerfully symbolizes her dedication to science under circumstances. Yet, her life was not without struggle. She faced gender bias, caste discrimination, and professional marginalization within the scientific establishment of her time. There were moments when her work was questioned, her credibility challenged, and her perseverance tested. But as her own words reveal, I refuse to be defeated. She stood her ground, guided by evidence, confidence, and self-belief. Invited by the then Prime Minister of India, Pandit Jawaharlal Nehru, to accept an assignment as a special officer to reorganize the Botanical Survey of India, she returned to India in 1951. From then on, besides the reorganization of the Botanical Survey of India, Dr. Ammal continued to be in the service of the Government of India in various capacities. From November 1970, she worked as an emeritus scientist at the Center for Advanced Study in Botany, University of Madras. She lived and worked in the center's field laboratory near Madras until her demise in February 1984. She was elected Fellow of the Indian Academy of Sciences in 1935 and the Indian National Science Academy in 1957. The Government of India also conferred the Padma Shri on her in 1957, and the Ministry of Environment and Forestry instituted the National Award of Taxonomy in her name in 2000. For the students of St. Teresa's College and the nearby colleges, her story is especially significant. Dr. Janaki Ammal chose a life devoted to learning, research, and service. This choice was radical in her time. Her life shows that each one of you has the right to define your path, whether it leads to science, arts, public service, entrepreneurship, or any other field. In her later years, Dr. Janaki Ammal became a passionate advocate for biodiversity conservation and indigenous knowledge systems. Her life tells us that science is not isolated from values. It is deeply connected to ethics, responsibility, and care for natural world. It is therefore, I believe, the most fitting that today's memorial lecture is delivered by someone who, in many ways, carries forward this legacy of scientific excellence, leadership, and inspiration for women in science. Professor Usha Vijayraghavan, who is a distinguished plant biologist, currently serving as the Dean of Division of Biological Sciences at the Indian Institute of Bengaluru. Her career rooted in rigorous science, international scholarship, and an unwavering commitment to mentoring the next generation. Equally important is her role as a leader and role model. Her journey demonstrates that women can excel not only at the laboratory bench, but also in shaping institutions. As we listen to today's memorial lecture, let us remember that Dr. Janaki Ammal's greatest legacy lies not only in her publications or awards, but in the example she set of intellectual courage, independence of thought, humility, and lifelong curiosity. Dear friends, may her life inspire you to ask questions fearlessly, to persist even when the odds seem stacked against you. As Dr. Janaki Ammal herself believed, my work is what will survive. May each of you find work and purpose that is worthy of your passion and perseverance. All the very best. Thank you.
Thank you, sir. May I request Ms. I.K. Nishida, Assistant Professor, Department of Botany and Center for Research, St. Teresa's College, to introduce a speaker for the day. Ma'am, please.
Thank you, Arya. Respected dignitaries on and off the stage, a warm good morning to all. It is my privilege to introduce today's distinguished resource person, Professor Usha Vijayraghavan, a renowned molecular biologist and one of India's most respected scientists in biological science. Professor Usha Vijayraghavan obtained her PhD from the California Institute of Technology, Caltech, Pasadena, USA, in the area of yeast genetics. She subsequently carried out her postdoctoral research at Caltech with Professor Elliot Meyrowitz, where she initiated her pioneering work on transcription factors regulating plant development, research that went on to shape her long-standing contributions to plant biology. She is currently professor in the Department of Microbiology and Cell Biology and Dean of the Division of Biological Sciences at the Indian Institute of Science, Bengaluru. Notably, Professor Vijayraghavan is the first woman to serve as Dean at IISC, a premier institution for science and engineering. She also holds the prestigious J.N. Tata Chair, one of only 3 endowed professorships at IISc. Since joining IISc in 1990, her research has focused on understanding how genes control flowering, plant morphology, and development using rice as a model system. Her research uses advanced genetic and genomic tools to understand key regulators of rice plant development. She has made landmark discoveries on transcription factor-driven gene networks that control plant and planicle architecture and floret fertility, significantly advancing our understanding of crop development and productivity. Her team has also made important contributions to understanding gene regulation at the post-transcriptional level, particularly pre-mRNA splicing. Professor Vijayraghavan is a fellow of the Indian National Science Academy and the Indian Academy of Sciences, and held the J.C. Bose National Fellowship for 10 years. She has mentored several doctoral students and has served the scientific community through editorial and advisory roles. Her work has been recognized by awards such as the National Bioscience Award of the Department of Biotechnology, the Harswaroop Medal of the Indian National Science Academy, the C.V. Raman Award of the Karnataka State Science and Technology Council, to mention a few. We are deeply honored to have you with us today, ma'am. Please join me in welcoming Professor Usha Vijayraghavan, an eminent scientist, equally admired as an enthusiastic and inspiring teacher, to deliver the prestigious Dr. E.K. Janaki Ammal Memorial Lecture. Ma'am, the stage is yours.
As we proceed, the dignitaries on the dais may kindly occupy their seats off the dais. We now come to the highlight of today's program, the E.K. Janaki Ammal Memorial Lecture. It is my honor to invite Professor Usha Vijayraghavan, Professor, Department of Microbiology and Cell Biology, Indian Institute of Science, Bengaluru, to deliver the memorial address. Please, ma'am. May I kindly request everyone to keep their mobile phones on silent mode to ensure the smooth conduct of the program.
Hello, can you hear me? Can everybody hear me? Great. So we can turn that off. That's good. Can I start? Good morning, everyone, and it's a great pleasure and privilege for me to be present today at St. Teresa's College. And I begin by first thanking the President of the Kerala Science Congress to think of me and associating whatever little bit I have done with the, India's iconic woman scientist, Janaki Ammal. Thank you, Professor Nair. I know that you could not be here today, but I would like to place that on record. Thank you also, Dr. Sabu, Member Secretary of the Kerala Science Congress, in making these fantastic arrangements. Dr. Anu Jacob, Principal of the College. I'm sure you are all daily experiencing her leadership in this very, very important college in Kerala, which has celebrated the centenary year. Thank you also, Dr. Nishita, who is the Chair of Botany, and I forget the faculty from zoology who I met, I forget her name now, and of course, all of you students who showed up over here to listen to me. It's simply wonderful to recall the contributions of Dr. Janaki Ammal. She is a legendary plant cytogeneticist and a plant breeder who was driven, as rightly said by Dr. Sabu, by curiosity. And curiosity, like Dr. Arjun said, not bounded by anything. Not bounded by, I want to achieve this medal, or I want to achieve that prize, or I want to do this. Bounded by only the scientific interest in pursuing the truth and understanding how nature works. That was Janaki Ammal. Regardless of whatever situation she was in, she did not lose her focus and her curiosity to pursue science to understand nature. Her book, The Chromosomal Atlas of Plants, covered in detail 10,000 species and described how their chromosomal content is organized. And this actually paved the way for what now in modern day one can go and follow up chromosomal studies by using molecular markers and so on and so forth. So this is a precursor of many things that can go on in plant genetics and genomics in the 21st century. I'm indeed, I repeat, indeed honored to be giving this memorial lecture in her name. In today's talk, I will try to cover some of the research work that we have done in trying to understand how the rice plant develops. But before I do that, I'm going to backtrack to take you because I know that this is a diverse student class, and some of you would have interests in different organisms, model organisms, in biotechnology, and biology, biochemistry, etc. So, I am going to take you back to what is the context, why do I study what I do, and why do I enthuse my team members in the lab to study what we do. So, let us go back billions of years in history. When we look at the origins of eukaryotic organisms, these are organisms where their cell is organized with a distinct nuclear component and cytoplasm, which contains many organelles, and therefore it partitions the specialized functions of the job into different organelles. The nucleus containing the hereditary material that is passed on from cell to cell and from generation to generation in a faithful way. That unicellular eukaryote was common to the plant and the animal kingdom and arose about 1.6 billion years ago. So, that is how ancient eukaryotic life systems are in Earth. And subsequently, so the origins of plants, you might be a zoology major, you may be a botany major, you may be a biotechnology major, it does not matter. But that tells you that several processes which occur in eukaryotic cells are likely to be common between plants and animals. The tools might be different, that is in the next slide. They then diverge to make these two distinct lineages, because one of the living beings in that many a billion years plus ago acquired a second organelle through the acquisition of a bacteria, which later on made a plastid, and this went on to give us the plant kingdom, right. So, while the broad rules, so now the question is, what can we understand about multicellular organisms, not the unicellular eukaryotes, multicellular organisms. How are these multicellular organisms put together to give you the constant reproducible shape development of that organism? If a mouse will always give you a mouse, it will never give you a fish will never give you a fly will never give you a nematode. Why? Why does a magnolia always give rise to magnolia? Janaki Ammal worked on magnolias a little bit. Why is it that sugarcane always gives you sugarcane and not rice? These are questions. How are they put together? What are the common features by which you can go from a single cell zygote represented by the fertilized egg? So, you always start with two gametes, the male gamete and the female gamete, an egg and a sperm, or an egg and a pollen nucleus if you were a botany student. That gives you the fertilized egg, and from this you get a spectacular complexity of tissues and organs that are made typical for that species, right. So, how do you go about doing that? So, it is very clear that one cell has to go to many cells, because the fertilized egg is a single cell. You have to generate more cells, and those cells should also, with time, as the organism is developing during embryonic development, cells have to get specialized, right. Second process, the cells have to talk to each other. The cells need to interact and communicate with each other such a way that they can organize themselves into a tissue structure with distinct identities, and cells can get specialized, right. And in animal systems, cells also move. For example, the cells on my face arise from the neural crest, which gives rise to my nervous system. So, the cells need not in animal development be at their final destination where they were made. They could have come from some other place. But this process of cell movement is very much restricted in plant development. Plant development mostly occurs through the first three processes. So, now we come back to the single cell. We have a single cell which duplicates, makes daughter cells, makes more cells by mitosis, and that is through the process of cell division that controls the faithful segregation of the DNA content. So, now all the cells are identical, they have the same inherited genome. How can you produce different identities for the cell? Why is it that one cell becomes the precursor for the neural tissues, another cell becomes a precursor for the mesoderm? In plants, one cell becomes a precursor for the shoot, another one becomes a precursor for the root. Why? How does that happen? That means that as the embryo is developing different tissues, regardless of what it may be, it has to turn on and turn off different sets of genes. And this is the problem thing of gene regulation. So let's imagine that you're in embryonic stage 1, gene product 1 is being made and the gene encodes, it could be a protein or it could be a non-coding RNA, whatever it is, and that can set the process of turning on gene 3, and therefore now you have a linear cascade which is an on-on cascade. If this is on, it will go and turn on this on, and when this is on, it goes on another on, so that you move from one step to the other and acquire new identities. So initially in time t0, only this product was present, but in time t2, both these products are present, and therefore this defines a stage that's different from this. Right? So this way one can progress in acquiring identity over developmental time as cells are dividing and moving along. Right? So now let's come to flowering plants, which is the topic that we are going to discuss in detail today. All those other 3-4 slides pertain to the development of any multicellular eukaryote. Right? So in flowering plants, what are they? Flowering plants, I'll show you in the next slide, are rather recent entrants to Earth, and they arose approximately the same time that there was a radiation of mammals. So mammalian systems and flowering plants in evolutionary time are approximately the same. So this is during the Jurassic end, the end of the Jurassic period. That's when flowering plants started blooming, moving all over, the dinosaurs were gone, and inconspicuous mammals, which were not even noticed, started to expand. So speciation of mammals and flowering plants and insects occurred at that era. But what is special about flowering plants is that the embryo is deeply embedded in maternal tissues. It's present within the seed. So the embryo is made within the seed by fertilization of the egg, which is present in the ovary, by a sperm nucleus which comes through the pollen tube. This then develops and gives you a very, very simple structure. A ball of cells, this is at the globular stage embryo, which is attached to the maternal seed tissue by a suspensor. And when you look at this embryo, there is very little information about what is going to happen later on. The only information that's present is this, what is called the apical basal axis. We know that this is going to be apex, this is going to be down, and these cells divide and give a later stage embryo where we have a small group of multipotent or pluripotent stem cells that give rise to all the above ground structures of the plant, and you have another group of multipotent stem cells over here that give rise to all the below ground tissues of the plant, which are not shown in this schematic. So nothing about the future of the plant's shape, organ system, nothing is present in the embryo. Contrast that with a mouse embryo. In all vertebrates, not only mammals, all vertebrates, embryonic development is when pattern is made. And almost every pattern of the tissue types are already laid down in the embryo. Post embryonic growth, that is when a chick hatches, a reptile hatches, a human being is born, or a mouse pup is born, they already have all the tissues. What you have is change in shape and change in size of the different organ systems. So I just told you that flowering plants and mammalian species evolved around the same time, but the mammalian embryo already at this stage will have everything. It will have an eye, it will have limbs, it will have a stomach, it will have liver, it will have kidneys, lungs, everything. So this contrasts very beautifully about how one can study development in plants continuously through the life of the plant, right. So on germination, it will, the above ground tissues, it will give you a stem, it will give you a leaf, it will give you an axillary meristem, and this mode of growth can continue. And when the time and place is appropriate, then this vegetative plant will start giving new structures, which are flowers that didn't exist before. So it is a miraculous model system for us to understand how plant development and organ systems can occur post-embryonically. We cannot study this in the mouse embryo. We have to go in utero to study all of this, right. So, therefore, this is a very, very exciting model to study development. Okay, now why is this not moving forward? Do you want to try and hit the forward button? Ah, there. Okay, maybe this battery is gone or something. Okay, so I come back to this slide which tells you the diversity of flowering plants. So, flowering plants exploded, and currently it is estimated that we have 350,000 species of flowering plants extant, forget about the ones which went extinct. So, it is a huge diversification that has occurred in this period of about 180 million years, and very early in the diversification of flowering plants is split into two lineages. One is a eukaryotic lineage, eudicot lineage, and the other one is a monocot lineage. And they produce a diversity of floral shapes. So, I told you that the plants will grow vegetatively, they will make a decision to go reproductive, and that means they have to start making flowers, and these flowering plants all produce flowers in many different ways. Where they are positioned in the body of the plant, how many floral organs they will have, all these things are vastly diverse, and this has fascinated human kind for millennia. People wrote poems on flowers, people bred them for making them beautiful for us. The rose that is cultivated, I do not know whether this is here, yes, you gave me a beautiful rose. That is not real, I mean that is not, that is manipulated by us, we created that rose. The wild rose has only 5 petals, 100, probably about 60 to 100 stamens and a carpel. So, of course, that is very nice, it is very fragrant, and the bees like it, and the birds like it, everybody likes it, but man wanted to take that and make it a little bit better and a little bit better. So, naturally that the cultivated rose is a mutant. Okay. So, this is how mankind has taken a few species and of plants and used them either for aesthetic purposes, for beautifying our lives, or for our nutritional nourishment, and we derive most of our caloric intake from a few species of plants, right. But if you want to understand flowering plants as a group and understand how do flowers get made, then you need a model system where you can deeply investigate how does genes, which I told you the genes need to be turned on and turned off, and how do cells acquire their identities. So, you need a model system where you can ask all of this, and that has greatly changed with the discovery of just a simple weed. It is just growing, it is a temperate weed found everywhere in the temperate zone, including in Kashmir, to Kazakhstan, to North America, to Europe, and that is a relative of the mustard family called Arabidopsis thaliana. And why did this become such a big deal? Arabidopsis became a model system because of its attributes. It had the smallest genome. So, its nuclear DNA content was the smallest amongst all flowering plant species. So, if you were to do Janaki Ammal type of work and look at the chromosome content by cytogenetics, you will see that there are only 5 pairs of chromosomes here. So, you can have 5 chromosome pairs, but you can be very, very large content. So, then you are not a model. So, not only do you have small number of chromosomes, the DNA content is also small, yet it is able to make all the typical organs of the flower that are seen in all other flowering plants. So, it still makes sepal, petal, stamen and carpel. It still makes flowers only after first growing vegetatively. So, therefore, one can now understand using the tools of molecular biology and genetics, how do you make such organs in the Arabidopsis flower, right. So, now about 30 years of deep research, genetics, genomics have given us a wealth of information on how it is that the floral organs are patterned, right. So I will just summarize that very quickly in the next 2 slides. So now this is a scanning electron micrograph. Okay. So technology is highly, highly empowering. So this is what I want to convey. So you don't get rooted or wedded to one technology. Get wedded to the problem. Get wedded to what is the biological question. And technology will keep on evolving, but keep your eyes and ears open to apply the right technology to answer your questions. So don't say that I'm a this technologist and I'm a that technologist. I mean, that's good, but don't get mired into the technology, understand the question. So if you look at the tip, growing apical tip of the Arabidopsis plant, you see a group of cells over here which don't look anything special. They just look like cells. They don't have any identity. They don't look like any organ primordia. These are the multipotent stem cells which are present in the tip of the plant. So all of you zoologists who are sitting in there, now there is big ha-hoo about animal stem cells and pluripotency and adult stem cells and tissue organization and organoids and all that in mammalians. That's all news for you people. For plant biologists, it's passé. All plant biologists knew that plants have meristems, and the meristems are the growth centers for the entire development of the plant. It is the descendants of these cells which get pushed to the side or the flanks, and this is called the peripheral zone of the meristem, from which organ primordia emerge, right. So this is an emerging primordia growing out as a bulge. That means cells are dividing rapidly over here, but they are not dividing here. They are growing rapidly here and creating this ball, but not dividing here. So how are these positioned? Why in the peripheral zone certain regions only give rise to the organ primordia, and that is why you will find diversity in the flowering plants. And then this ball of cells, which does not look very much different from this, then goes on to make new structures which are coming in concentric rings. So there is a ring of new structures. If you peel that inside, there is a ring of another new structures, then a ring of another new structures, and the central dome then grows out into a tube, and ultimately you get this beautiful flower. And this flower has precisely 4 sepals, 4 petals, 6 stamens, and a central carpel with a stigma. The first flower which is made by the Arabidopsis plant will look like this. The 100th flower will also look like this. The 1000th generation will look like this, and whether you grow the Arabidopsis in Ernakulam, in Kochi, in Bangalore or in Trivandrum or in Munich or in California, it will always look like this. Why? How? What? Those are the big questions. So it's very clear that you have to go from a group of cells which have multiple potencies. They can make 4 different organ types, and gradually in those regions you start defining which region is going to become sepal. So you have to turn on the sepal development genes in the right time and space. If you turn on the genes that control the formation of the sepal over here, it's not good, right. So it is precisely trying to, I think you have to hit the button, for some reason the forward is not working, yeah. So we want to, if you want to understand why this stereotypic pattern, then next slide, next, and what did the giants in the field do? Elliot Meyerowitz, who is my postdoctoral mentor, Enrico Cohen at the John Innes Institute, the very institute that Janaki Ammal also worked in, Heinz Sadler at the Max Planck Institute, and Martin Corneef at the Wageningen in Netherlands. These 4 people said that let's just begin with a clean slate. We don't know anything. Nobody knows how these 4 organs are made and why this stereotypic pattern is there. So they said let's make mutants, and they did it in 3 different species. 3 of them did it in Arabidopsis, 1 did in Petunia, another did in Snapdragon, and they got similar looking mutants. What was the question? The question was, why do we have this pattern, sepal, petal, stamen and carpel? So if you make a mutant in which this pattern is abrogated, pattern is gone, then that mutation is in a gene that creates this pattern, right. So shown here is an example of one such mutant in which the flower cannot or does not produce stamens and carpel. 2 organ systems are not being made, and instead in their place are made additional petals and additional sepals, and the organ system number keeps on increasing. So, this defined the agamous gene as the gene which is required for both stamen and carpel development, right. So, what was the premise? You do not need to know anything, you need to be able to think about the question and see what is the tool which is going to help me to identify the key players. So, that led to the discovery of agamous. I am not showing you other etc. And here is a case where actually the mutant is prettier than the wild type. So, that is your wild type we went in the previous one. Why is it prettier? Because it has got more petals, and that is what all of us want, right. So, this is really the mutant rose, the mutant camellia, the mutant azalea and all of that. So, those occurred naturally in the breeder, then kept on propagating them. So, agamous is one such mutant, and several other mutants were got. Sorry, I have to ask you to hit, yeah. And several other mutants were obtained, and not only did they obtain single mutants, they also obtained double mutants and put these double mutants in combination with triple mutants to decipher what is the code by means of which you can always make a sepal on the outer edge of the growing floral meristem and so on. So, that gave the code as a set of now what we recognize as 5 different factors. It started with 3, it was the know your ABCs, and now it's know your ABCDEs. Your alphabet soup, which gives you predictably the floral development, right. What is that alphabet soup? That alphabet soup is that wherever a class A gene is expressed and also a class E gene, this bar is now extended over here, you will always get a sepal. Wherever a class A gene and a class B gene and a class E gene is expressed, you will get a petal. Wherever a class B gene, a class C gene, a class E gene is expressed, you will get a stamen. Wherever a C and a E is expressed, you will get a carpel, and within the carpel where you get C, D and E, you will get a ovule, right. So, this is like setting up a zip code. Imagine, I think Ernakulam is a sort of linear city as I was driving through, so also is Chennai, but imagine Delhi or Bangalore. It is a very circular city. So, imagine setting up concentric zip codes, saying this zip code means you are in the airport and Yelahanka and the periphery, then this zip code and this zip code and this zip code. So, the combinatorial action defines these 4 different regions within the growing meristem, and therefore gives you these organ types positioned in the right way. This is very good. But what are these ABCDE alphabet genes which I told you about? How do they go about doing their job? To know that, you have to go deeper, which means you have to get to molecular biology and biochemistry. So, you can start with biology, embryology, whatever, genetics, but you also have to be capable to do the other things, right. So, they then went on to find out where these mutations reside on the chromosome, cloned the gene, found out the equivalent of the wild type, that is the normal, complemented it and discovered that all of these genes encode transcription factors, meaning these are proteins that are master switches because they can identify their downstream genes where they need to bind and turn it on. So, imagine I told you that embryonic stage 1, embryonic stage 2, what are these matchbox genes? They are one of the blue dots or the green dots, which can go and find where they want to activate downstream gene programs. So, these are transcription factors which are proteins that make various complexes, and these complexes direct the downstream program. Next slide please. Right. And what is that downstream program? These ABCDE in various combinations are going to give you this organ structure. So, I also teach developmental biology to the IISC undergraduates. So, I tell them that look, it depends, it might be a vanilla ice cream with cherry on top, a vanilla ice cream with chocolate sauce, a vanilla ice cream with gems or sprinkles on it, but the outcome is different. Your tongue tastes it differently, your strawberry ice cream will taste different. So you can make different combinations of the same thing. You have a cone, you have a ice cream, and you have some sprinkles. But what you will feel will be different. That's what's happening over here. You make these combinations which can control the form. Very nice. So 30 years of plus research in Arabidopsis, and I did my postdoctoral work in plant biology. So like Nishita and Sabu and all were mentioning, my foray into plant biology is as a postdoctoral fellow. So I wasn't born knowing that I was going to do this or I'm going to do that. But whatever you choose, you have to simply be driven by passion, and that passion will make you excel. There's no doubt about it. My bachelor's is in zoology honors from Delhi University with subsidiary of botany and chemistry, but I took additional subjects. They allowed me to take additional subjects because I also liked physics and I also liked mathematics. So I have a BSc honors zoology with botany, chemistry and additional subjects, and I was a gold medalist there. But that didn't say that I'm a zoologist for life. So I said, okay, this is all very nice, and now is coming the age when people should understand molecules, this, that and the other. And I did my master's in biochemistry at the Postgraduate Institute for Medical Education and Research, because I really thought that we should be working in the border between chemistry and biology and genetics. My PhD was in genetics using the unicellular eukaryote yeast, and my postdoc is in plant biology. Okay, so that vastness of experience I think has stood me in good stead, but I did not, it's not like I planned to do all of this. At each step you have to challenge yourself to see what is it that I want to do and go for the top place of that, prepare for it. I didn't put my career timeline. I usually do this in a career talk, and things happen and things will happen. Good things will happen if you try hard. Some things don't happen, let it be, forget. But at every stage in your career, challenge yourself to go that extra mile, and the career path opens up. There's no doubt about it. It will open up. I did not plan to be the first dean of the Indian Institute of Science, first woman dean. I just did whatever I had to do, and it happened. So one of the questions that inspired me when I came back to India, I started initially doing Arabidopsis work, but the relevance of the problem, I wanted to go beyond that, right? So if we can understand how flowers develop in Arabidopsis, can we take that knowledge or that lessons and apply it to a crop plant, right? And crops are hard to work with because many of them are often polyploid species, the genetics is difficult, molecular tools were not available, nothing. So at that time the rice genome size got measured. Okay, like chromosomal content, we all knew how many pairs of chromosomes in rice, but its genome turned out to be the smallest amongst all cereal crops. It's the smallest, compare it to sorghum, compare it to maize, compare it to millets, compared to sugarcane, very complex genome, it was the smallest. So if you have a small genome, then you can do a lot of molecular biology because you can get to the genes that control the process. So I said, okay, why not rice? That doesn't mean that I knew anything about rice, but having decided that I am going to study rice floral development, I have to learn everything about it. I was an assistant professor, so those of you who are assistant professors, this is not the end. Make a beginning every day. I knew nothing about it, nothing other than eating rice. Okay. I went to the agricultural college and went to the field. I mean, I had seen the rice fields and all. My grandfather from Tanjavur, I have seen plenty of rice fields, but never I had bothered to go and pick it out and look at it. So we went, pulled out the plant, looked at the development and found there's a millions of questions we can ask, millions. You just have to cross that energy barrier. So you open the flower and you see that when you take it out, they are minuscule flowers. The flowers are very tiny and the outer two organs are called the lemma and the palea, and they don't look like the sepals. They always close the flower. the rice and all grass flowers are always closed, because, and in order to see the inner structures, you have to peel it open, and then internally you don't find a ring of petals, you see two small fleshy organs on one side, and you see a stamen and you see a carpel. So, some things are same, some things are looking different. What is different? The lemma, the palea and the lodicule. The first, these organs are looking very different than that of Arabidopsis and other eutychod plants. So, then we went backwards. I said, okay, where are the flowers coming from? So, this is, instead of an SEM, I have shown a drawing. This is the shoot apical meristem of a young seedling. It has those pluripotent stem cells and it is making leaf primordia. And when the time is right, then this entire apex starts growing like a cone. And in that cone, it makes primordia which are going to give rise to new cones. And those new cones are making some structures over there, and these are called branch primordia. These are primary branch primordia, secondary branch primordia. Finally, one short branch is made, which is called a spikelet, in which there are two pairs of bracts, and then comes the lemma, palea, lodicule, stamen and carpel. So, this is what the rice floret looks like. So, rice floret is made after a complex set of branching of the meristem. So, in Arabidopsis, directly from the flank, this would be a flower, this would be a flower, this would be a flower, this. But in rice that does not happen. You generate more meristems and you generate more meristems, primary and secondary meristems, which then give rise to the flower. So, many new questions can be asked. How do you get this complexity of branching? And that complexity of branching determines how many flowers will be present. If you have less branching, you will have less flowers. And this is species specific and variety specific. There are varieties which branch very highly, there are varieties which branch less. So, you can ask what is driving the branching. You can also ask questions on what is driving this organ development, and are these organs homologous or similar to dicot floral organs. So, these are questions that you can ask just simply by looking at how the rice flower is made, when it is made, what is made. Next slide please. So, you do not have to, now these are more detailed SEM etc. to convince you that the floral organ, it is completely closed, this is a cross section, it is not an open. And these positions of these organs, the types of cells that they are making and their homology to dicot flowers is questionable. Next. Yeah. So, I told you about the alphabet soup A, B, C, D, E, right. So, those are shown over here in different colors and their combination gives you this structures. So, are those genes present in rice? So, we discovered in Arabidopsis, which is a flowering plant, you know what are those genes which are controlling Arabidopsis flower development. Do their homologous genes occur in rice? Yes, they do. Our work as well as that from several Japanese, Chinese, Australian labs discovered these homologs. But very interestingly, many times there is gene duplication and diversification. For example, in Arabidopsis, the class E genes shown here by yellow color is represented by 4 genes. All these 4 genes have only one function, that is they have to partner with the other genes and any one of them can do the job. Why does Arabidopsis have 4 of them? Duplication, genes got duplicated, but they are functionally the same. So, having one of them is good enough, and that is why unless you make a quadruple mutant, that is you mutate in Arabidopsis all the 4 sepalata genes, you will not lose class E function, and if you do not lose class E function, then you will get a normal flower. But in that quadruple mutant which is not having any class E function, all the organs of the flower are replaced by leaf like structures, right. But now you come and look at rice. In rice, we find that there are 3 rice genes which are related to one of the Arabidopsis sepalata 4 gene. So, why does rice have 3 genes related to this, but it is also branched off because there are many genes over here. All of these are seen in only other cereals. There is maize and there is sorghum here, but not in other dicots. Why do we have this radiation of related genes in grasses. So that suggested that maybe these genes are related to the Arabidopsis genes, but their functions are unique, and that's what we found. In rice, the single class E gene, there are 5 class E genes, if mutating only one of them can change the floral development. So this is the normal wild type rice. This is a mutant which was isolated by a Korean group. This is an RNA interference transgenic that we made in my group, and this is a group which was made by a Chinese group. In all of them, mutation or abrogating one gene is enough to spoil everything about the rice flower development. And instead of making a normal flower, you are getting these bract-like structures continuously repeating, or you get mosaic organs and so on. So that's a gold mine. You have a mutation in one gene and the identity of the flower is gone. Floral organs are not being made. So we then asked, where is this gene expressed? To do that, we have to use a method called RNA in situ hybridization, that is, in the tissue, in tissue sections, we find out which cells are expressing the RNA from our gene of interest. So you make antisense RNA synthetically and add it onto the slide and find out which cells have that mRNA to which it can hybridize and will give the signal, right. So this is a growing floral meristem, and all the purple region is expressing OSMADS1. So this is a floral meristem before any organ primordia are made. So if you are a master regulator, that switch should come on before the slave. So this is the master, it comes on first and it turns on the identity of this group of cells as floral, and this master itself gets regulated by other masters. So nobody is a prima donna, everybody is important in their own time and space, right. So this master is now shut off from the other regions, it is present little bit over here and it is present over here, and later development it is helping in the organ development over here. So that means we know that the single gene mutation which changes the identity of the rice flower, so the flower can no longer make any floral organs, is itself controlled in time and space, right. So we asked, what does it control? Instead of asking how it is controlled, we said, okay, let us see who does it control and how does it control. For that, we did 2 different methods. Next slide, please. Yeah. So this whole thing is just about less than a centimeter, and in that are developing at least 50 flowers. Each flower is this, just this. So minuscule amount of tissue. So we went and dissected and collected for 3 to 4 months. If you collect the tissue, you will get enough material to make one RNA prep. So nothing is easy, but the goal makes it all worth it, right. So what did we do? We collected this tissue from panicles which are at 0.2 centimeter to 2 centimeter. 0.2 centimeter is how much? 2 millimeter. Okay, dissect all this from fields and go to the field, pull out the material, bring it to the lab, dissect it under the microscope, flash freeze it, keep on doing this, collect enough material, and we made chromatin from them. What is chromatin? Chromatin is the DNA which is already wrapped with the histones, right. So we made the chromatin, and wrapped on the chromosome along the chromatin will be the transcription factor. Transcription factor will be sitting on top of the gene who it is going to turn on or who it is going to turn off. And one of those transcription factors is our transcription factor, OSMADS1. So from that chromatin, we then pulled down the chromatin to which our transcription factor is bound. For which, how do we pull it down? We made an antibody. Now that antibody means I have to go to rabbit. Okay. So all zoology and all very good. I said, okay, we don't have to wait. We will make the protein in bacteria, we will inject into rabbit, we will give booster shots, and we will isolate the antibody which is reactive to our transcription factor which came from rice. All the tools are there. Okay. So we raised the antibody in rabbits, purified it against the plant protein and use those antibodies to find out in which chromatin regions is our transcription factor bound. Okay. So, the rice genome has about 35,000 genes. Which genes is it binding to and turning on? And we did that analysis and found that it binds to about 3,000 different genes, and its binding positions are near the transcription start site, and sometimes even within the gene in intronic regions, and sometimes at the 3' end. And this also analysis means what? You also have to do bioinformatics. You have to map the binding sequence. You have to understand what is the DNA sequence at the binding site. You have to know how to work with free energies. So, you collaborate with people and you say, okay, this is the data set I have, this is the type of question I want, can we apply your tools? So, collaborating with others allows you to understand your data to the depth that you will not be able to do individually. So, we identified that the MADS1 transcription factor binds to any DNA sequence which has this type of consensus element, but is also binding to other regions, new regions which we called as A track. And these are defined by looking at the free energy of the bound versus the unbound sites. So, what did we discover? We discovered a transcription factor whose activation is required for making a floral meristem. We defined that that transcription factor binds to about 3,500 genes in the rice genome, and it changes their expression. What does it do change expression? Does it always turn on genes, or does it turn off genes? Does it do both? So, for that what we did was we did transcriptomics. So, we did a mutant. We made a mutant in OSMADS1 by knocking it out, by doing a deletion of the whole gene. So, this is genome engineering. So, when this was done, I collaborated with Veluthambi and Madurai Kamaraj University. We made this mutant. So, we compared in the entire flowers of the wild type versus the mutant, what all genes are expressed wrongly. In the mutant, you get a flower which does not have any organ types, everything looks like a bract, right. So, something has gone wrong, some wrong genes are being expressed, or the right genes did not get expressed, both can happen. So, we did differential NGS and determined how many genes are deregulated in different developmental stages to understand that OSMADS1 is activating in early floral development this about 1,200 genes. It is repressing about this. So, what is it doing? When early flowers are being made, it is activating these specific 1,200 genes. So, that defines their identity as floral. In later development, it is activating about 250 genes, this about that, in order to make organ differentiation. So, at different times it is turning on different switches, but not only is it turning on different switches, it is also turning off other switches. So, you can see here the red. So, while it is turning on 1200 genes, it is turning off 400 genes. So, in time and space, it is like a conductor of a symphony. It is saying on, off, on here, off here, this, that, and that symphony gives you the flower, right. So, you need to understand the regulator from the point of view of something that controls development. So, using this information, we are now building gene regulatory networks. So, we know now that in very young floral meristem, when the organ primordia still not come out, this is a floral meristem and it is not a leaf primordia, not a branch meristem, anything. It is activating these genes, it is activating this, it is activating this, and this is a feed forward loop. This feed forward loop reinforces each other. So, you all can now discuss and find out what you learned. You can feed information to each other and reinforce yourself on what it is that you want to take away from this lecture. But later in development, it switches, it turns off these very same genes, such a way that now it can promote the emergence of organs. So, the same factor can be an on switch or an off switch depending on what time you are looking at it, right. So very, very interesting new questions are coming from this. So it's such a complicated network, 3,500 genes, and that network is controlled in time and space. You massage it a little bit here, it will go this way, you massage this way, it will go that way. And now we have all the tools to start digging deeper and deeper into this. We can unravel one hub and see what happens. Is it redundant with the other hub? So many different new questions can come, because now we have a set of 3,500 genes that you can play with, not 35,000 genes. Right? So I'll tell you a little bit of one more story, only 5 minutes to just tell you that every gene offers you new possibilities. So this is another gene which belongs to the class B, and class B genes are required to make petals in the second whorl and stamens in the third whorl. That's what we know from Arabidopsis, from tomato, from petunia. Now what about in rice, right? So we took a class B gene and asked, which is one of the class B genes is called pistillata, and we asked whether this pistillata clade of genes, are they functioning to make the petal-like organs and the stamen-like organs in the rice flour. So we knew in Arabidopsis, class B genes are required for making petals and stamens. So are they doing the same thing in rice? So what is the petal equivalent? Very interesting little organ. I told you it's called the lodicule. It's present in the base of the flower, and it is asymmetric because it's not present in a ring. You can see the ring of stamens, but you see only 2 lodicules. This lodicule swells and pushes open the lemma and the palea around the time that the stamens are coming out, and that allows for fertilization, and then it closes back in and allows the seed development. So the lodicules are very specialized petals whose position, anatomy and function is different than that of petals. So what is the class B gene doing? How it is making this new structure? It's a class B gene. So we asked the question by looking at the pistillata genes in rice, and we found that the pistillata genes are 2 of them given in yellow over here, MADS2 and MADS4, and when you knock out both of them, you can see the flower does not have any lodicule, just makes massive amounts of carpels. No stamens, no lodicules, just carpels getting repeated, because you have only C function gene and A function gene. B has been removed. But if you lose only one of them, like over here you lost MADS4, you see that the stamens are coming, they are kind of all right, they are kind of fertile, but the lodicule is thin and long. When you lose the other one, the lodicule is normal, but the stamens are abnormal. So looks like the 2 relatives, these are cousins, both are pistillata-like genes, but they have divided their function. One is more important for lodicule or petal development, rice petal, other more important for stamen development. But we also discovered one more thing, that when we knock out both of them, there is a phenotype of parthenocarpy. The ovule is expanding even though there is no embryo inside it. So the pistillata genes, which are supposed to control petal and stamen development in rice, are also controlling carpel development, female fertility. So if we had not taken rice as a model, we would have not made that discovery. Okay. So this tells us that you can take highly conserved transcription factors which are present from Arabidopsis to rice. So conserved over 120 million years. The proteins look similar to each other, but how they partner with other transcription factors can allow it to give different outcomes. And we also discovered that factors can acquire new functions. In rice, we discovered that MADS2 is controlling flowering time. That is, when you go from making leaves to making flowers, it is controlling this flowering time, which it does not do in Arabidopsis. So genes can acquire new functions, genes can duplicate and share functions, and this you will discover only by going into different model systems. Right? So this controls flowering time, and together they control parthenocarpy. Both of these are phenotypes not done by Arabidopsis. So I must tell that I have been blessed by having I think I've guided about 27 PhD students and about 20, 25 postdocs. I've been blessed by being able to sell my inspiration and my ideas. Every time it's a different idea. It's a tall benchmark, and all of them have generously learnt and given their time. These are all the PhD students and postdocs who have been with me. I think I might have missed some 4 or 5 of them simply because I've been around for quite some time. I've had collaborators. I continue to collaborate with Uli Grossniklos, who's at Switzerland, Velutambi at Madurai Kamaraj, Manju Bansal at IISC, Binay Panda, who is now at JNU, Arjit Samal, who is at in Chennai Mathematical Institute, Sundaresan, who is at UC Davis. At different times, depending upon what the question required, you talk to people, you make them interested in your problem, and you collaborate with them. I've been generously supported by the Indian Institute of Science, Department of Biotechnology, the Science and Technology also. I'm sorry, I forgot to mention over here one important funding body, which is actually another reason why I chose to work with rice as a model 20 years ago. That's the Rockefeller Foundation. So Rockefeller Foundation, I came back to India after my postdoc, and I've taken up position. I'm working on Arabidopsis. Arabidopsis has absolutely, it is fundamental knowledge, no immediate use to any mankind, it's not edible, whatever. And I was approaching various funding bodies for funding my research. So they said, okay, no, we won't fund Arabidopsis research, but we do fund plant biotechnology. If it is in rice, we will fund, but those are all breeders and all that, they don't do molecular biology and all. So if you want to do molecular biology with rice, we may consider. So I wrote a compelling proposal, and it is because of that funding support from Rockefeller Foundation, because of my prior training, my teachers, my mentors, and the courage that you have to take on a topic in which you have never worked before, and it opened a rich set of questions for us to ask. So Rockefeller Foundation, my teachers, my mentors, and the canvas that I had, do what you want at the Indian Institute of Science, but you have to achieve. You can't say that I'm going to do this, that and other and not deliver. You have to keep on delivering, you have to take people along with you and make everyone succeed. More than half of my students are professors in various places. You might encounter them, and those who are still with me will go on to accomplish a lot. I wish you all a brilliant journey in your undergraduation, post-graduation, and stay motivated. There's fantastic career waiting for you in science. You have to set the benchmark and you can achieve it. Nothing is achieved easily. So be ready to put your worth and you will achieve it. Thank you all.
Thank you, ma'am, for that insightful and thought-provoking lecture.
We will now have a short interaction and question answer session. The floor is now open for questions. May I request the participants to keep their questions concise. You can raise your hands, we'll come with the mic.
Too much information.
Any questions?
My name is my name is Parvati Hari. I'm a third year BSc Zoology student. So, what do you do if you have a varied field of interest? Like, for example, if you have an interest in marine biology while also having an interest in a completely different field, how do you select a topic to continue research in?
What is this completely different topic?
For example, if a person has an interest in marine biology,
No problem.
but having an interest in something completely different like agriculture, for example, how do you select between the two?
So you don't, so you don't need to select, because you can look for marine models which are informative in maintaining the marine ecosystem. So you said agriculture, so that means it has to do something with plants, right?
Yes.
Or it could have something to do with prawn or whatever. So if you want to study that, you can do some research on, for example, seaweed. Right?
Yes.
Okay, so you can look at how does seaweed develop. You can look at algal species. You can look at one of the models in alga are brown algae fucus. So there are
Yes.
many model systems which are very, very contemporary right now, because agriculture and farming of the sea is something for the future. But these are all difficult from the genome perspective, but technology is coming. So now you can employ technology to go forward. So I would suggest that you look at how people are studying algal biology, plankton biology, and all of this will link your diverse interests.
Thank you, ma'am.
Yeah.
Hi ma'am, I'm Alina from MSc Zoology. My question is, you already asked about the alphabetic transcription factors. If one transcription factor is disrupted, can the others compensate for its function?
Very nice question. So, a very nice question. So, this is exactly what is happening in Arabidopsis. So, one of the transcription factors is the class E sepalata gene is represented by 4 members. Okay. So, so the class E gene is not just E, it's E1, E2, E3, E4. All 4 genes are making exactly the same product. So, they do compensate for each other. So, if in Arabidopsis you make mutation only in E4, the flower is still normal. You make mutation in E4 and E3 and E1, and only then flower is disrupted. So, there they do compensate. But in rice, we found that E genes are represented by 5 of them, E1, E2, E3, E4, E5. But only one of them is enough to abrogate the development. So, in evolution, they can acquire subspecialization. That is species specific. So, in Arabidopsis, they are redundant. They can substitute for each other. But in rice, in the 120 million years, these 5, imagine that you have quintuplets. The quintuplets can be identical, can be not identical. Over time, they can evolve. So, that is what has happened in rice. So, they cannot fully compensate for each other. Each one has become important in its own right.
Thank you, ma'am.
Yeah.
Ma'am, how might the climate change affect the transcriptional regulation of flowering plants?
Very nice. So, so far we have been focusing on how genes control the process of development, right. So, we are talking about transcription factors which can switch genes on and off and give you the development. The 2 transcription factors which I talked about are working with flower development. So, we in order for making fertile male and female gametes, the temperature, the growth temperature is very important, right. So, even a fluctuation of 2 degrees is enough to give pollen infertility. That means pollen development genes are particularly sensitive to the change in temperature. So, what are those genes in pollen development which are abrogated because of temperature change are all topics which we have to discover. What we know is, if we talk about climate change, there are 2 critical windows in plant development. One is early seedling development, when the seedling is still making leaf primordia. If the temperature shoots up, then it affects the rate at which new leaves are coming out, right. Many of those steps involve the control of leaf primordia, photosynthetic capability, etc. So how do transcription factors get controlled, influence that? Possibly because when there is a stress, the plant entire physiology is diverted to combat that particular stress. So when you have high temperature, you usually will be having high amount of oxidative stress. You will also be having a high amount of respiratory burst stress. So you will be making antioxidant proteins, you will be shifting your metabolism to handle that stress, and the impact of that is development is slowed down, right? That is one. Second thing is many of the climate induced changes can also be epigenetic. So what do transcription factors do? They turn on a gene, like I said, or turn it off. But epigenetic factors are the ones which maintain the status. Once you turn on a gene, how are you going to keep it on forever? For that, there are chromatin modifiers. So climate change can not only influence through transcription factors, it can also influence through epigenetics. And in plants, typically these are the reproductive phases are very sensitive to climate change, and early seedling development very sensitive to climate change.
Hi ma'am, my question was how does the mutation can affect the genomic level of rice floret development in the recent timeline?
Yeah. So because the transcription factor is activating specific genes in the genome. In the genome, let's say the rice genome, there are about 35,000 genes, right? So in order to make a particular floral organ, you don't need all the 35,000 genes. You need only specific set of them. So that set of genes which it is controlling, we identify through methodologies like chip sequencing, differential transcriptome analysis, etc. So the current 21st century tools are what allowed us to identify what genes does OS-MATS1 control when flowers are being made. So I told you that it's activating 1,200 genes. How did I define those 1,500 genes? I just didn't make the bar chart. It's based on the data that it is those genes whose expression is controlled by the binding of OS-MATS1 to that particular locus. So in the current scenario, we can define which exact genes are being controlled by our transcription factor using the tools of genomics.
Thank you, ma'am.
Any other questions? So one announcement, those who have not registered on paper, outside, not the QR, on paper, they are requested to register before they leave. It's for the certification process. Okay. Any questions? Is it done? Ma'am.
Okay, thank you.
Ma'am, one more question, ma'am, from my side. Ma'am, I'm Varada from Zoology Department, first PG. So, ma'am, based on your findings, what do you think that it is the most promising direction for future research in this field?
There are hundreds of directions. So, what I would say if you're a zoology major, so maybe you're interested in developmental biology?
Yes.
Right. So if you're interested in developmental biology, you would want to try to understand how patterns are formed and how patterns are maintained, right? So why is it that if you're interested in, let's say, vertebrate development, why is it that an organ has a particular size and shape? It is defined by the primordial stem cells, which will divide and control. But why is it that that organ size is of that size only? Why is the liver only this big? Why is it not bigger than what it is? What is the relationship of the size of different organs and organ development? These are all big challenging questions. So how do you balance? Why is our one arm and the other arm same size? Got it? Why doesn't my one arm touch the ground? another arm be at my elbow. So somehow there is an organismal level, system level control of organ size and in development. So these are big questions that one can ask. One can also ask what are the role of genes and epigenetics in controlling these processes. These are all questions which are very pertinent from a development point of view. From the point of view of diseases and so on, one can look at genomes wherever there is huge diversity like the human genome or species diversity genomes where are done. How do genetic variations in the gene contribute to a function? Right? Why is it that butterflies have wings of this shape and size, whereas moths have a wing of this shape and size? All of this you can discover by employing the tools of genetics and genomics. Right?
Thank you, ma'am.
Yeah.
Thank you all for the engaging discussion. May I now invite our esteemed dignitaries to join Professor Usha on the stage. I invite Dr. C. Arunan, Senior Principal Scientist, KSCSTT, Sri Arjun Prasad, Scientist, KSCSTE, Ms. I.K. Nishida, Assistant Professor, Department of Botany and Center for Research, St. Teresa's College, Dr. Himasri P.R., Junior Scientist, KSCSTE.
As a token of appreciation, we now invite Professor A. Sabu, Member Secretary, KSCSTE, to present a memento. A small correction. So as a token of appreciation, we now invite Dr. C. Arunan Sir, Senior Principal Scientist, KSCSTE, to present a memento to Professor Usha Vijayraghavan. Please, sir.
Thank you. To formally conclude the proceedings, I invite Dr. Himasri P.R., Junior Scientist, KSCSTE, National Transportation Planning and Research Center, to deliver the vote of thanks. Ma'am, please.
Respected dignitaries on and off the dais, esteemed guests, ladies and gentlemen. It's my honor to propose the vote of thanks at the conclusion of the E.K. Janaki Ammal Memorial Lecture organized by KSCST in association with St. Teresa's College, Ernakulam, as a prelude to the 38th Kerala Science Congress themed Celebrating Science, Transforming Lives. Today's program stands as a fitting tribute to Dr. E.K. Janaki Ammal, a pioneering Indian scientist whose path-breaking contributions to plant genetics and cytogenetics laid a strong foundation for modern scientific research in India and contribute to inspire generations, especially women in science. I sincerely thank Dr. Anu Joseph, Principal St. Teresa's College, Kochi, for the gracious hospitality and institutional support extended for hosting this distinguished academic event. I express my deep appreciation to Professor A. Sabu, Member Secretary, KSCST, for his valuable presidential address and for his continued leadership in strengthening science and research outreach in Kerala. I greatly acknowledge Sri Arjun Prasad, scientist, KSCST, for his insightful introduction to the life and legacy of Dr. E.K. Janaki Ammal and Ms. I.K. Nishita, Assistant Professor, St. Teresa's College, for elegantly introducing the speaker. We are deeply honored by the presence of Dr. Usha Vijayraghavan, Professor, Department of Microbiology and Cell Biology, Indian Institute of Science, Bengaluru. We thank her sincerely for delivering an inspiring and intellectually enriching memorial lecture which beautifully reflected the scientific excellence, depth, and societal relevance that Dr. E.K. Janaki Ammal stood for. I also commend St. Teresa's College, the organizing committee, faculty members, staff, and student volunteers for the seamless and efficient conduct of this program. I am pleased to note that this year marks the Golden Jubilee of KSCST, National Transportation Planning and Research Center, NATPAC, making this occasion even more meaningful and reaffirming our collective commitment to advancing science, technology, and research for societal transformation. I extend my sincere thanks to KSCST, NATPAC, St. Albert's College, St. Teresa's College, and to the audience for your attentive participation. Thank you.
Thank you, ma'am. I once again thank all the dignitaries on the dais for your presence. Before we disperse, a few important announcements. E-certificates will be sent to your registered email IDs. Kindly scan the QR code displayed at the registration counter outside. And also, I repeat, make sure you registered it on paper. Not only the QR code, make sure you wrote it on paper. For updates on the 38th Kerala Science Congress, please follow our official social media handles and visit the KSC website. We encourage all participants to complete their registration at the earliest. I repeat, please do your registration at the earliest. Thank you.
Thank you all once again for your presence and active participation. Wishing you a pleasant day ahead. The third UG and second